All right, so today we're going to do a deep dive into something pretty amazing, secondary antibodies.
Oh, yeah.
These tiny little things, they have this huge impact on scientific research.
Hmm.
And you sent over some information
hmm
um, about choosing the right secondary antibodies. That sounds
Yeah,
like you want to really understand how to pick the right one.
definitely. Yeah. There's a lot of factors to consider there.
Absolutely. And what's really cool is just how widespread these secondary antibodies are.
Yeah.
They're used in so many techniques.
Yeah.
You think about Western blots, ELISA,
hmm
immunohistochemistry,
Yeah.
even immunofluorescence.
Right.
And it's like anywhere you're trying to visualize and really zero in on a molecule, these things are essential.
Absolutely. That's a great point.
And your source goes into all these fascinating details, like matching the antibody to the species it came from,
Right.
or the different ways to actually make them visible.
Mhm.
Some of them even glow. It's wild.
Yeah. It's pretty cool.
And then it really emphasizes how the choices you make there can seriously impact your research findings.
Oh, yeah, absolutely. I mean, choosing the right secondary antibody can make or break your experiment. It's not just about seeing something, it's about getting reliable data,
Right.
making sure your results are actually accurate. I've seen really, really well-designed experiments just totally go off the rail
Oh, wow.
because of a poor antibody choice. It's kind of heartbreaking when that happens.
Yeah, I bet. So, okay, so before we get into all these little details, can you just break it down for us? What exactly are these secondary antibodies? How do they actually work alongside those primary antibodies?
Sure. So basically think of it this way. You're searching for a specific protein, let's say, in a cell,
Mm.
okay?
Okay.
And your primary antibody that's designed to bind to that protein
Yeah.
very specifically, like a lock and key, right?
Right.
But the key itself, it doesn't light up. You need a way to actually see that it's found its target.
Right.
And that's where the secondary antibody comes in.
Gotcha. Okay.
The secondary antibody,
Yeah.
it binds to that primary antibody,
Mm-hmm.
and it carries a signal, basically a beacon. So that could be a fluorescent molecule that lights up under a microscope, for example,
Okay.
or an enzyme that creates a visible color change. These are what we call detection methods.
Okay, so there's multiple ways to actually see what's going on, at that tiny level.
Yeah, exactly.
Your source talked about, these different types of beacons. What are some of the common ones researchers use?
So on the fluorescent side, there are things like FITC, TIITC,
Hmm.
and those ELISA fluor dyes. You've probably heard of those.
Yeah.
Those are popular for immunofluorescence, when you want to actually see that protein glowing.
Right.
And then you have enzymes like HRP and AP.
Okay.
And these are used in what we call colorimetric detection, basically. It's creating a visible color change that you can then measure, used a lot in ELISA, for example.
Oh, interesting. Okay.
Oh.
So much variety. It's pretty amazing. Now, the source, it really emphasized how important the host species is when you're picking a secondary antibody.
Mm-hmm.
And they gave this example. If your primary antibody was raised in a rabbit,
Right.
then you need an anti-rabbit secondary antibody.
Right.
Can you break down why that matching is so crucial? Why does it have to be that specific?
Sure. So basically, each species, they produce antibodies that have slightly different structures. It's
yeah.
like trying to use a key from one brand of lock on a totally different brand. It just won't work.
Right.
So if you don't match that secondary antibody to the species of the primary antibody, it's not going to bind, and you won't get any signal at all.
Ah, okay. So it's all about compatibility.
Exactly.
Making sure that they can actually connect.
Yeah.
Now, speaking of connections, the source also mentioned this thing called cross-reactivity, which I guess you don't want,
Right.
especially in techniques like IHC and IF.
Yeah, definitely.
Can you explain what that is and why it's such a big deal?
So cross-reactivity, that's like your secondary antibody getting distracted.
Mm.
Ideally, it should only be binding to that primary antibody.
Right.
But sometimes it might bind to other molecules in your sample. And those might have similar structures, so that can lead to false positive results.
Oh, so it's like it's giving you a signal,
Right.
but it's not the signal you're looking for.
Exactly, yeah. And that can be a real problem, especially in techniques like IHC and IF,
Mm-hmm.
where you're looking at these complex tissues. There's a lot of different molecules in there.
Right, right.
So that's why it's important to choose antibodies that are specifically what we call absorbed against other species.
Okay.
That helps minimize these unwanted interactions.
So it's like you're fine-tuning the specificity of your secondary antibody
Sure.
to make sure
Yeah.
it stays focused on the target.
Yeah.
Okay, now the source also went into this thing called conjugation,
Mm-hmm.
and it listed all these different tags,
Right.
HRP, AP, FITC, TIITC, ELISA fluoride dyes.
Right, yeah.
So what is conjugation exactly, and why so many different options?
Well, conjugation is basically attaching that beacon to the secondary antibody.
Okay.
The tags that you mentioned, the HRP, the AP, the fluorescent dyes, those are all different types of conjugates.
Okay.
And the one you choose depends on your detection method.
So if you're using a fluorescence microscope,
Right.
you need an antibody that's conjugated to one of those fluorescent dyes.
Exactly, yeah.
And if you're using a method where it's based on color change,
Yeah.
you need an enzyme conjugate.
Exactly right. You've got to match the tool to the task.
Yeah.
And each one has its own pros and cons.
Oh, I bet.
It's going to impact the sensitivity, the signal stability, and how well it works under certain experimental conditions.
So there's like a lot to think about even within that one choice of conjugation.
Oh, yeah. Oh, sure.
Okay. The source also talked about affinity and specificity, which seem to go together.
Mm-hmm.
Can you unpack those for us a little bit? Like, what do those terms mean when we're talking about secondary antibodies?
Sure. So affinity, that refers to how strongly the secondary antibody binds to its target. In this case, the primary antibody. So a high affinity antibody, that's going to bind really tightly, which is going to give you a strong signal.
Makes sense.
And specificity, that's making sure it binds only to the target
Mm-hmm.
and not to anything else in the sample.
Okay, so high affinity means strong signal. High specificity means like a really clean signal
Exactly.
without a lot of background noise.
Yeah, you want both. That's the ideal scenario.
Yeah, I can see why both are important.
And there's a technique we use called affinity purification that plays a big role here.
Okay.
Basically, it removes any antibodies that don't bind strongly. So you're basically making sure that the secondary antibody you're using is really good at its job.
Okay, so it's like filtering out the impurities to get a
Yeah.
more concentrated
Yeah,
and reliable product.
right.
Okay, so far we've talked mostly about the antibody itself,
Right.
but your source also mentions how the actual experimental conditions themselves can impact the antibody you pick.
Oh, yeah, absolutely.
They specifically mentioned Western blots
Yeah.
and IHCIF as examples.
Uh-huh.
So can you unpack that a little bit for us?
Sure. So each technique has its quirks.
Right.
In Western blots, for example, the size of the secondary antibody can actually affect how the protein moves through the gel.
Oh, wow.
If it's too large, it can kind of create drag. Too small, it might not bind effectively.
Wow, so it's like the physical properties of the antibody
Yeah.
come into play.
Right.
Okay, and what about IHC and IF?
Well, with IHC and IF, you're working with tissues, right?
Right.
Which are much more complex than, say, just cells growing in a dish.
Mm-hmm.
And so things like the type of tissue, the
Right.
way it was prepared, that can all impact how accessible your target molecule is.
Okay.
Some antibodies might be better at getting into certain types of tissues, or they might work better with certain preparation methods.
Wow.
It just depends.
That is fascinating. Like all these things are intertwined.
Yeah.
The antibody itself, the conjugate, the conditions of the experiment. You need like a holistic understanding to make good decisions.
That's a really great way to put it. And, there are some general guidelines,
Right.
but the best choice often comes down to experience and really knowing your technique and your antibodies.
Right.
Yeah.
Well, it's a lot to think about. Wow. It's amazing how many choices are involved just to pick the right secondary antibody.
Yeah.
I mean, you got the host species,
Mm.
the conjugation affinity specificity.
And don't forget about validating your choices with those control experiments.
Right, the controls.
Those are critical to making sure your results are accurate.
Ah, yeah. The source did mention that, but I'd love to hear a little bit more about it.
Sure.
Can you explain, like, why are those controls so essential when you're working with these secondary antibodies?
Well, basically, controls help you rule out any results that might be misleading, or that are just straight up false positives.
Okay.
Remember, secondary antibodies are supposed to bind to the primary antibody, right? Which then binds to your target molecule. But what if that secondary antibody starts binding to something else
Oh.
in your sample, and you get a signal that doesn't really reflect your target at all?
Okay, so it's like a check to make sure the signal is actually coming from
Right.
the interaction that you're actually studying.
Exactly, yeah. A very common control is to just run the experiment without the primary antibody.
Okay.
And if you're still getting a signal from that secondary antibody, it's binding nonspecifically, and you got to figure out why.
Okay, that's a really clear way to think about it.
Yeah.
It's like you eliminate a variable to see if it's really necessary.
Mm-hmm.
Okay. Are there other types of controls that people commonly use with secondary antibodies?
Oh, yeah, absolutely. The specific ones will depend on the technique.
Mm-hmm.
But for instance, in Western blots, it's common to use a loading control.
Okay.
That helps make sure you've loaded equal amounts of protein in each lane of your gel.
Right.
So any differences you see are really due to your target protein levels.
Okay, yeah, so you're really controlling for the amount of sample
Right.
to make sure you're comparing things fairly.
Yeah, exactly.
That makes a lot of sense.
It's all about having those safeguards in place to make sure your findings are solid.
Right.
I've seen researchers spend weeks, sometimes even months, troubleshooting an experiment.
Oh, no.
And all they needed was a simple control experiment to point them in the right direction from the beginning.
Oh, wow.
Yeah, it can be frustrating.
It really shows how important careful planning is.
Absolutely.
Having a deep understanding of all the ins and outs of your technique.
It makes all the difference. Yeah, choosing the right secondary antibody and using the right controls, those are key to reliable research.
Okay, now I'm curious, with all your experience in this field, have you ever encountered any surprising or unexpected results when working with these secondary antibodies?
Oh, countless times. That's what's so fun about research, right?
Right.
Pushing the boundaries, and sometimes you just stumble on something totally unexpected.
Can you share an example?
Sure. So one that comes to mind is this experiment we were doing with a fluorescent secondary antibody.
Okay.
We wanted to visualize this protein inside a cell.
Mm-hmm.
And we had this hypothesis about where it should be, But when we actually looked under the microscope,
Yeah.
the signal was totally different from what we were expecting.
Oh, wow. What did you see?
Well, instead of being in this one specific area, the protein was spread out all over the cell,
Oh, wow.
almost as if it was being transported along this pathway we hadn't even considered before.
That's so interesting.
It was a total surprise, but it opened up this whole new research direction for us.
That is so cool.
Yeah, it just goes to show even a basic experiment can lead to new discoveries,
Right.
especially when you have the right tools.
It really highlights that power of being able to actually see what's going on.
Absolutely, and how the choice of secondary antibodies shapes what you can learn about the system.
It's like you get a new lens to view the world with all these hidden details become visible. So, okay, we've covered a lot of ground here. Selecting secondary antibodies, those controls, even the element of surprise in research.
Yeah, it's been a great conversation.
What are some key takeaways that you would want our listener to remember?
Well, I think the most important thing is to really appreciate how complex these seemingly simple tools are.
Right.
Choosing a secondary antibody that's not a trivial decision, it can really make or break your research.
Yeah. It's about understanding all those little details, the host species, the conjugate, all of that.
Yeah, and how all of that relates to your specific technique.
And like you were saying earlier, don't underestimate those controls.
Absolutely.
They can really help make sure your results are accurate and reliable.
It's about avoiding those false signals that can derail your whole project.
And I love what you said about staying open to the unexpected.
It's all about the journey.
Sometimes the most exciting findings come when you least expect it.
Yeah, science is all about exploration.
Absolutely. Now, before we wrap up our deep dive, is there anything else about secondary antibodies that you think is worth touching on?
There's one more thing I think we should talk about.
Okay, let's hear it.
Okay.
So you said there was one more thing about the secondary antibodies that we should talk about.
Yeah. We've talked about all the technical stuff, how important those controls are, even, the surprising results you can get.
Yeah.
But there's this whole other area underneath all of that, and that's how the antibodies are actually made.
Right. Yeah, the source didn't really get into that.
Yeah.
But I know most antibodies are made using animals.
Right, exactly. It's a pretty well-established process. You basically introduce your target molecule, what we call the antigen, into an animal like a rabbit or a mouse.
Okay.
And their immune system, it responds by producing antibodies against that antigen.
Right.
And then you can extract those antibodies and purify them for research.
It's a really powerful technique, but I can see how that raises some questions.
Yeah.
Especially as research is kind of evolving.
For sure, there's a growing movement now towards developing methods that don't use animals at all.
Yeah, I've heard a little bit about that.
Yeah.
What are some of the approaches that people are trying?
So one that's really promising is called phage display.
Oh.
Instead of using an animal, you basically create this library of antibodies in the lab.
Okay, so in vitro.
Exactly, in vitro.
Gotcha.
And then you screen those antibodies against your target. And the ones that bind really well, those are the ones that you select and produce in larger quantities.
So it's like an artificial immune system
Yeah, that's a great way to think about it.
in a test tube.
Yeah, and it completely bypasses using animals, which is great for a lot of reasons.
Right. I can see that both ethically
Yeah.
and also just in terms of refining that whole production process.
Absolutely. And there are other methods out there too,
Oh, really?
like using cell-free systems or even synthetic antibody libraries.
Wow.
It's a really hot area of research right now.
Yeah.
Could really change the way we produce and use antibodies in the future.
That's amazing.
Yeah.
It sounds like we're at like a turning point in this field,
Yeah.
driven both by new technologies, but also just like a changing view of how we do research.
Yeah, I think that's a great point.
It's really exciting.
It is. And it makes me wonder, what if we're just scratching the surface here?
What do you mean?
What if there are totally new ways to see these molecules, methods we haven't even thought of yet?
Okay, that's a really interesting question.
Like maybe there are these undiscovered molecules or particles out there that could interact with our targets and make them visible in completely new ways.
It's like a scientific conspiracy theory.
Kinda, yeah.
There's this whole hidden world that we could reveal
Right.
if we had the right tools or, just shifted our perspective a little bit.
Exactly. And who knows, maybe our listener will be the one to make that breakthrough.
That's a great challenge to leave our listener with.
Yeah.
Well, it's been awesome exploring the world of secondary antibodies with you today.
Yeah, it's been a great conversation.
We've covered so much the technical details, the bigger picture for research, and even, gotten a little glimpse into the future of this field.
Yeah, for sure. It's been a pleasure talking with you and with our listener.
Yeah,
Hopefully this sparks some more questions, and a desire to keep learning.
absolutely.
That's what the science is all about.
Well said. And to our listener, thank you for joining us on this deep dive.
Yeah, thanks for listening.
We hope you learned some new things and maybe even got inspired to dive a little deeper into the scientific world.
Yeah.
Until next time.